Thanks, Jeff, and thanks for the invitation to give a talk. I'm going to be talking about a relatively new technique using stable isotopes that has I think has traditionally been, at least the power of the technique has been underutilized by marine scientists anyway. And there's a couple of good reasons for that, but I'm just going to give you a couple of quick vignettes in terms of how this technique might be used to address some pretty fundamental questions in coral reef ecology. And the first one is going to be looking at that blue highway that a couple of people have already shown, the movement of snappers, at least certainly snappers, other species as well, from nearshore estuary from nearshore areas to offshore reefs. And the second one is going to be even more fundamental, I think, in looking at Darwin's paradox in terms of how do coral reefs living in such oligotrophic waters have such high biodiversity and high productivity? And I think both those questions are actually addressable using this technique. So as I said, the technique is not new. It's been around for at least 20 years, but it's really the application of the technique that has been somewhat limited. And so it's compound-specific analyses of stable isotopes. We're going to focus on amino acids in this particular talk. And the reason why I think it hasn't been widely adopted is that the challenge is that it's not analytically easy to do. It takes a reasonable amount of time. It's much easier to send a bulk stable isotope analysis to UC Davis. In a couple of months you have the data back. That's not what you can do with these kinds of analyses. So bulk data has been used for a long time, and ecologists are very familiar with it. Unfortunately, they're not as familiar with some of the limitations of the technique, and we'll get to that in a little bit. But the problem, in a nutshell, is that if you have a bulk sample of, say, mussel, you have a number of different amino acids, and they're each fractionating differently. So when you do a bulk sample, you're basically integrating over all those different fractionation factors, and there's actually a lot of different things that can cause differences in that fractionation, which will then be seen in terms of the isotope data. So that's a big problem that we can actually overcome by using this new technique. And it also means that bulk analyses, because of all those different things that could be influencing fractionation, that bulk analyses are actually remarkably difficult to interpret if you are careful, and that becomes a particular problem on places like coral reefs where we have very complex food webs to start with. And again, this technique is a way of getting around at least some of those problems. So we get around those problems using this compound-specific analysis. I'm not going to go into any great detail in terms of the analysis itself, but basically what we do is this. We use a gas chromatogram to separate those amino acids in time. And so this just shows a chromatograph. So each of those amino acids are actually coming off the column at different times. And as they come off the column, we then send them to a combustion furnace. They get converted into CO2, and then we suck them into the mass spec and get our, in this case, carbon-13 to carbon-12 ratio. And again, that's a process that takes on the order of 1 hour per sample as opposed to a couple of minutes if you're doing bulk analysis. So it's certainly not for every application. It is time consuming. But what we've been doing in our lab is trying to scale up these analyses to get at ecologically meaningful sampling to use this technique to address some of these questions. This just shows the results of a feeding experiment with a number of different amino acids. This was very controlled. We knew exactly what the diet was. We read the fish. And the only point that I want you to concentrate on is these are all the non-essential amino acids. These are amino acids that we can actually synthesize ourselves, that fish can synthesize. So as you can see, there's a lot of fractionation and a lot of variability in the fractionation of these amino acids that we're producing ourselves, and that's simply a function of fractionation during that synthesis process. What's more interesting for us is these amino acids here. These are amino acids that we can't synthesize ourselves, and neither can any higher animals. So these are basically propagate unfractionated through food webs. So these give us a very good idea of the carbon isotope values at the base of the food webs that animals are feeding at. So there is no fractionation, and this makes them in some sense much more easy to interpret because we don't have to worry about that variable fractionation generated by a number of different things. So from now on, we're going to focus simply on these amino acids, the essential amino acids where we can know that we don't have to worry about that fractionation. And they're simply going to propagate through the food web with an isotope characteristic signature of the base of the particular primary producer that has fixed that carbon. And that's what we'll do from here on for both the examples. Okay, so you've seen this graph already a couple of times today, the schematic. What we do know is that we find a lot of juvenile snappers, for instance, in nearshore waters, and we find adults on offshore reefs. And it's been very convenient to imagine this as some sort of blue highway where these juvenile snappers are moving out of these nearshore areas and onto these offshore reefs through time. It's actually proved remarkably difficult to actually quantify that movement and to see if perhaps we have in fact captured the entire story by this schematic. And so we decided to test this in the Red Sea using a very common snapper, Lucianus ehrenbergii, that's widely distributed throughout the Indo-West Pacific. And in this case, we sampled at a site just south of Jeddah in Al-Leith. And what we're going to do is we're going to look at samples that are collected from nearshore habitats, both inshore seagrass, mangrove areas. Here's coastal reefs. The shelf break is about here. All these reefs here are actually on the shelf. We have one continental island. And then we have 4 reefs that are actually off the continental shelf. So these are actually, again, those very steep oligotrophic reefs that Michael was showing you earlier. The color coding is going to be the same throughout, but it's a little bit much to keep track of, but we'll do our best. This just shows if you do counts on these reefs, this is what you see. As we go from coastal wetlands out to these offshore reefs in terms of the size frequency, this is exactly the kind of data that people have used to infer that juvenile snappers are residing in these nearshore coastal areas, the small guys right here, and then moving out into these offshore habitats as they get older. So this is basically is exactly the prediction that we would make of this blue highway movement of small snappers. So the first thing we needed to do was to actually generate the isotope signatures that we were going to use to classify individual fish back to those nearshore habitats. And this just shows you, these again all muscle samples from Lucianus ehrenbergii, and this just shows that we can actually separate these different habitats based on what is now a multivariate signature, but it only uses those 5 essential amino acids that we know are going to propagate unfractionated through those food webs. So here we have coastal wetlands, very different from shelf reefs, from those oceanic reefs, coastal reefs here, and actually the continental island, Abu Lat, actually is separate from all those as well, and something actually quite interesting is going on Abu Lat. But anyway, the take-home messages that with this technique, we were able to pretty much uniquely identify each of these different habitats using this compound-specific approach. So the next thing was, okay, so how are we going to actually retrospectively determine where an adult snapper may have been residing as a juvenile? And we'd actually, in our lab, done a lot of chemistry looking at the inorganic aragonite chemistry of otoliths, which has been known for a long time as actually a pretty good way of tracing the movement of fishes through isotopically or elementally different water masses. It turns out that was the easy part because otoliths are about, 95 to 98% aragonite. With this technique, we actually had to be a lot more careful, and it was a lot more difficult because now we were going after the 2 or 3% of the otolith that's protein with the amino acids that we were going to use. So the amino acids in the otolith, and in particular in the core region of the otolith here, which represented the area that was laid down when that fish was a juvenile, by looking at the isotopic composition of the amino acids in that part of the otolith, we could actually use maximum likelihood to estimate where exactly those fish were coming from. And this is what the data looks like. I'm just going to walk you through it. I'm going to have a whole lot of different graphs coming here, but this just is a schematic of our study area. We have Abu Lat, we have these offshore islands, we have these nearshore habitats, and these coastal reefs. So for the reefs closest to those nearshore habitats, it's not too surprising. About 75% of the adults on those coastal reefs did indeed have the signature. This pointer. So here we go. There were a few, about 25%, that appeared actually not to be using those nearshore habitats, and we assume that those guys are actually just simply settling directly onto those coastal reefs. They actually don't seem to need those habitats. If we go out to these 16-kilometer reefs, and you can see again there's a significant proportion of adult snappers on those reefs that have made the movement of that 16 kilometers from these nearshore habitats to those offshore reefs. But again, you can see that although most of them, some of them are actually doing that, we have others that are again appearing to settle directly onto the reefs without going through those nursery habitats at all. If we come out one more, now we're actually at 32-kilometer reefs out here. And now what's interesting is that we still do get some coastal wetland fish, although they're obviously not nearly as many. What we do see is that a number of fishes at this distance are actually residing as juveniles on this continental island. So the seascape configuration of this particular area is actually having a pretty interesting effect on the migration or the movement pathways of these juvenile snappers. And finally, what was really interesting to us is that if you actually now go all the way offshore, now we're off the shelf completely on these oceanic reefs off here, and now we see a completely different picture. Now we actually don't get any fish that have come from those nearshore habitats. So now no fish actually that we've ever analyzed has made the movement from here all the way across the shelf to these outer habitats. The majority of fish now are actually settling directly onto those offshore reefs without any coastal nursery phase, and some of them are actually coming from Abulat Island. So the juveniles that actually are settling here, they seem to be the ones that in some sense I don't know, brave enough, whatever, to make that dash across open water to these reefs here, but none of the fish from closer inshore appear to be willing to make the leap off the shelf to those oceanic reefs. So again, it really emphasizes the impact that seascape configuration has on the movements of these juvenile snapper species. So what we have then, and just in summary, is we have fish that seem to be quite happy making at least a 30-kilometer plus movement from these nursery habitats out to these offshore reefs. And we have some other snappers that if they settle, if they're already in some sense offshore snapper, by settling onto this island, they too can make a generation, they can move about 25 kilometers from the island off to these offshore reefs. But again, we don't see, this is the migratory break, if you like, right here. We don't see snappers from these shelf habitats making it all the way out to these oceanic reefs. The limitation of the technique, obviously, is that we can't actually tell exactly how the fish are getting there. The fish may be making a straight directed movement from these coastal habitats, say, out to these outer shelf reefs in this case. That may simply be a directed movement straight off the bat. Or they may be doing something like this, where they actually, in fact, step their way across different reefs, maybe perhaps through Arbolet Island to these reefs here. Again, we haven't got the resolution in terms of being able to sample the otolith at sufficiently small scales to get some idea of which particular pathway they may be using. But it's certainly an area that we're developing to try and get that sample size requirement as small as we possibly can. Okay, so that's the first vignette. The second vignette goes to the question of, in some sense, Darwin's paradox, right? How are reefs able to maintain such high productivity and such high biodiversity in what are traditionally thought of as oligotrophic waters? And so this is sampling that we did, and we're going to look at reefs that we sampled, in this case, a more food web analysis, a number of different species at 10-kilometer distances, 20-kilometer distances, 30 and 40-kilometer distances. So we're going to go basically cross-shelf to the edge of the shelf, and then we're actually going to go off the shelf as well. And as you make that transition, you actually go through some pretty different habitats as well from these. This is, in fact, a nearshore coastal reef, and you can already see the water isn't particularly clear, pretty good coral cover, lots of particulate matter in the water. As you get further offshore, you get structures like this. Oops. So now we have clearer water, certainly, really interesting columnar structures. And then if we go all the way offshore, we have these more typical Red Sea reefs, very steep sides, typically very clear water. So that's the gradient that we're moving across in the following diagrams as we go from 10 kilometers all the way to 40 kilometers across the shelf. And so the first thing we needed to do then really was to be able to characterize the isotopic composition of the important end members at the base of those carbon food webs. And so what those 4 carbon end members that we chose to look at was phytoplankton, macroalgae, acroporid corals, and detritus. And for detritus, it's actually kind of tricky to get a sample that you might classify as detritus, but we use sea cucumbers in this case. So the first thing to do was to collect a number of different samples from each of these 4 end members and to characterize the isotopic composition of those end members. And this is a 3-dimensional representation of that data. Remember, we have 5 amino acids. We're just going to show them basically in 3 dimensions using a very simple, straightforward principal component analysis. And this just shows you that we actually get very good separation among each of our 4 isotopic end members. It doesn't look like phytoplankton and macroalgae are that different, but it's actually just the graph itself. They really are quite well separated in multidimensional space. So remember, these essential amino acids don't fractionate as they move up the food web. So we can actually use these signatures to say, for instance, with a mesopredator, we can use that Lujanid species again, how much of the carbon in that mesopredator has come from each of these 4 different end members at the base of the food web. And so these are the species that we chose for our food web study. We have with our top predator was actually a moray. We have mesopredators, planktivores, grazers, corallivores, and a detritivore. And I should say that this was what we thought we were dealing with in terms of each of those different feeding guilds. So really we're able to ask the question, for instance, with Tinochetus, is Tinochetus really a detritivore, in which case it would basically have 100% of the carbon in the muscle of that fish would have the unique signature of detritus, right? So how does it look? I'm just going to bring these. I'm going to use one distance just to give you a feel for the data, and I'm going to bring each of the species into our 3-dimensional plot, which is basically going to place those different individuals, each dot as an individual, in this multivariate space. So Stegastes, close to macroalgae, coral, Chaetodon trifascialis was certainly closest to the coral. Amblygyphytodon should be a planktivore, and it was actually closest to that phytoplankton end member. Tinochetus was actually pretty variable, but you could perhaps make the argument that at least some of the carbon was detritus. Sordidus is thought to be a grazer. It looks to me, first, and we'll get to the data in a little bit, looks much more like a detritivore. We have Lucanus ehrenbergi, sort of mesocarnivore, plotting reasonably close to sort of in between macroalgae and phytoplankton. And the moray eel, actually, as we'll see in a little bit, plotting almost completely on top of phytoplankton. So what we're going to do is we're going to use a Bayesian mixing model approach to get at actually the relative contributions of each of those 4 end members to the muscle tissue in each of those different fish. And this is how we're going to go. So again, the graphs are going to be very similar. We start off at the nearshore reefs, A, B, C, D, and then getting out, 10, 20, 30, 40 kilometers at these offshore reefs. And so what we can see right off the bat here, this is supposed to be a farming damselfish, and indeed, at least on the inshore sites, a lot of the carbon, all the way through actually, the majority of carbon certainly does come from the algae that it farms, but there's also significant contributions from plankton and from detritus. So it's not quite as easy. That's certainly not all it's doing is eating the algae that it's grazing. It's actually getting plenty of carbon from other sources as well. This is Amblygyphytodon indicus. This should be a planktivore, and indeed what you can see if we go to the offshore, very oligotrophic waters, there we see 80% are indeed, but that actually changes once you get into these more productive, less visibility, more particulates in the water column, and you can see that, in fact, now it's almost 50-50 in this case between plankton and algae. So on those reefs with more particulates in the water column, it's obviously picking up organic matter out of the water column along with the plankton. So it's not simply a planktivore. This is Chloroasordidus, and again, this is supposed to be a roaming grazer, but I think it's pretty clear from these data that it looks much more like a detritivore than it does look like a herbivore, all the way across. Tinochetus is supposed to be a detritivore, and indeed, there certainly is plenty of detrital carbon in Tinochetus, but there's certainly other things as well. It's obviously eating significant amounts of algae, especially on these inshore reefs where there is indeed more macroalgae for the fish to be feeding on. This is Trifascialis, Chaetodon trifascialis, a presumed obligate corallivore. And on these inshore reefs with pretty high coral covers, it does indeed seem to be eating primarily coral. But as we get offshore on these very steep-sided oligotrophic reefs, there's obviously a significant detrital component in that as well. Lucianus ehrenbergi is a good example of just how variable that those different compositions can be as you go through space. So now we go at the inshore reefs. It's actually almost, it's an algal signature in that carbon, and so that's probably from herbivorous invertebrates that it's eating. But as we go offshore, we can see that actually the phytoplankton component becomes much more important. So again, it's not feeding on the phytoplankton or zooplankton, but it's feeding on presumably on fish and invertebrates that are feeding in that new production phytoplankton food web. And this one was actually kind of particularly interesting to us. This is the moray, which turns out to be not an indiscriminate eater of fish at all, but a very, very specific, but targeting very specific species or groups of species that are planktivores. It's pretty easy if you go to these reefs. There's a ton of chromis, there's a ton of anthias on these reefs, and the morays all the way across the shelf seem to be targeting these planktivores in terms of the fish they're eating. They don't seem to be eating scarids, they don't seem to be eating a whole bunch of species. They really seem to be focused on what are very abundant planktivores at each of those sites. Okay, so just to wrap it up, so this, again, it's not a new technique, but what we are doing is applying it to situations where it typically hasn't been applied to before, and it provides a really interesting and tracer, both for assessing connectivity in terms of the snapper example, and in terms of looking at the structure of coral reef food webs. We've talked about carbon in this case. In terms of the snappers, so we do get certainly plenty of fish that are making 20 to 30 to 35 kilometer movements across the shelf from nearshore habitats out to these mid and outer shelf reefs. And so, yeah, that's obviously they're doing that. That blue highway for those species is certainly for those individuals is certainly very important. But there are certainly plenty of other snappers that don't take that highway, that seem to be able to settle directly onto those reefs. And it begs the question, between us, we spend a lot of time on these reefs, so where are they? How come we're not seeing these little guys? And I think there's probably at least two potential answers. And one is that these little guys are actually too deep, that they're settling into areas that we simply don't typically get to using scuba surveys. And the other answer is that they are just incredibly cryptic, that during the daytime when we're typically out on these reefs, they are deep in the reef matrix and hiding presumably from predators, hopefully not from us. But I think those are at least two examples, at least two possibilities, but it does make you think that we probably should be not relying completely on visual surveys to get at this kind of information for these species. And I think, so reef food webs are complex. I don't think that's of any surprise to anyone. And I think for us, our challenge is actually to see if there are any general principles for food web functioning. And we're starting primarily at the moment and looking at relatively pristine reefs, going to reefs where we still have a full suite of apex predators, for instance, and trying to quantify the food web structure using this approach. We've talked about carbon, but you can do very similar analyses for nitrogen as well. So in some sense, the carbon data is going to tell you so which particular food webs in terms of those primary producers the fish are relying on. Nitrogen is going to tell us something about the structure of those food webs, how those food webs are actually organized. And I think that together, that's actually going to be a very, very powerful analysis. What we're interested in doing after that is actually seeing if there are particular ways that human impacts actually disturb those food webs. And I think we're hopeful that perhaps there's actually relatively simple things that humans might be doing to avoid the detrimental impacts that humans seem to have on reefs, even at very low human densities. So I'm out of time. I'll leave it there. Thank you.